Hello, today we're going to talk about Chapter 7, Control of Microbial Growth. So let's start with a little history. Ignaz Semmelweis and Joseph Lister were instrumental in setting the stage for microbial control methods, and they did most of their work in the mid-1800s. Before this, before the widespread application of aseptic surgical techniques, about 10% of all deaths were due to nosocomial infections or hospital-acquired infections. Delivering mothers, their risk was as high as 25%. And during the American Civil War, a surgeon may have cleaned a scalpel by wiping it on the sole of his boot between making cuts. So let's talk about some terminology. Sterilization is removing all microbial life. Prions are highly resistant to all forms of sterilization. Remember, prions are infectious proteins. They're not living, but they can still cause infection and disease. The definition of sterilization implies the absence of prions. So if something's sterile, there's absolutely no microbial life or prions present. Disinfection is removing pathogens from surfaces or substances. And antisepsis is removing pathogens from living tissue. You'll need to know these terms for the test. All right, some physical methods for microbial control. So what are some things you can do to control microbes? And one of the things we do is modifying temperature. So moist heat sterilization can denature proteins. And this is one of the most common ways that we preserve food, by canning, right? An autoclave is like a pressure cooker on steroids. We use them in both a laboratory and medical settings to clean equipment, media, glassware, anything that can handle the heat and pressure. And the pressure and heat are intense. We're talking like 15 pounds of pressure per square inch and 121 degrees Celsius. It kills all organisms and endospores, but not those infectious proteins, prions. We can also use dry heat sterilization, and this kills by oxidation. We can use this like when we flame our loops in the lab, or incinerating things, or using hot air as a sterilant. Pasteurization is another way that we use temperature to control microbes. It reduces spoilage organisms and pathogens from food or laboratory solutions. So it's high temperature for a short time. So, for example, milk at the grocery store is treated at 72 degrees Celsius for 15 seconds. And you can also get ultra-pasteurized dairy products in the grocery store. These are some organic milks or like Fairlife, and a lot of creamers and heavy whipping cream have been ultra-pasteurized. Those go to 140 degrees Celsius for less than a second. And they generally have longer shelf lives. All right, we can also use low temperature to inhibit and slow microbial growth. Of course, we're all familiar with this. Refrigeration or deep freezing, if we keep the temperatures low, it slows the rate of growth of microbes, kind of puts them on pause. All right, another way that we can control microbes is by filtration. We use HEPA filters to filter air, and that removes microbes that are greater than 0.3 microns or 300 nanometers. Membrane filtration removes microbes from liquids, and that can be anything larger than 0.22 microns. Viruses range in size from 5 to 300 nanometers. So HEPA filters will not be effective at removing most viruses from the air. The influenza virus is 125 to 150 nanometers in diameter. And the rhinovirus, which causes the common cold, is only 18 to 28 nanometers. All right, and this is an example of sterilizing a liquid by filtration. So you put your sample in the upper container, put a cap on it. At the bottom of that upper container, there's a membrane filter with an extremely tiny pore size. And you can choose different size filters based on what you need to remove from your sample. There's a vacuum line that's going to suck the air out of the bottom container. And that vacuum is what forces the sample through the filter and down into the collection container at the bottom, where you have your sterile filtrate. Radiation is another way that we can control microbes. Ionizing radiation like X-rays and gamma rays damage DNA, and low-level radiation has been approved for use in the U.S. on spices, meats, and vegetables. Disposable dental and medical supplies, such as plastic syringes and surgical gloves, are treated with radiation, and the Postal Service actually uses electron beams to sterilize certain classes of mail. Non-ionizing radiation like UV light also damages DNA, but it doesn't penetrate. It's only good for surface applications. Sometimes we use UV light in the lab to clean safety glasses or micropipetters, different things like this that might have DNA contaminant on the surface. Microwaves kill by heat, so the radiation itself isn't particularly antimicrobial, but you can use it to raise the heat of a solution or a substance up hot enough to effectively pasteurize. So I use a microwave at home in my kitchen to heat milk up above 160 degrees when I make like yogurt or kefir with milk for my goats at home. Of course, you have to make sure that you're stirring things adequately because microwaves don't heat uniformly. So there will be hot spots and cooler spots. You need to make sure you're stirring. All right, so here's some examples of non-irradiated and irradiated strawberries. And it makes a difference in shelf life. All right, let's talk now about chemical methods of microbial control. So apart from things we can do, like modifying temperature or radiation, what are some chemicals we can use to control microbes? So there's some different actions of microbial control agents. Some alter membrane permeability, and that damage results in the cell contents leaking from the cells. So if we can destroy the cell membrane, we can destroy the cell. Some microbial control agents damage proteins. If we can damage the proteins, like little bags of enzymes, enzymes are folded proteins. And if we unfold them, or we change their structure due to chemical treatments, it makes those enzymes not functional. All right, and thirdly, some microbial control agents do damage nucleic acids. Heat, radiation, and chemicals can all be lethal to cells. All right, so this figure is decreasing order of resistance of microorganisms to chemical biocides. So the most resistant microbes are prions, those infectious proteins. They're not living. The next most resistant are endospores of bacteria. Under that are mycobacteria, cysts of protozoa, vegetative protozoa, gram-negative bacteria, fungi, including most fungal spores, viruses without envelopes, gram-positive bacteria, and viruses with lipid envelopes are the least resistant to chemical biocides. Some microbial characteristics and control things to consider here are that gram-negative bacteria are more resistant. They're harder to kill than gram-positive bacteria. Pseudomonas are highly resistant to biocides. Their outer membrane provide them protection, and those porins are highly selective of which molecules they permit to even enter the cell, so they help protect those gram-negative cells. Mycobacterium tuberculosis has mycolic acid and a waxy lipid component in the cell wall that resists the effects of these chemicals. It helps block them out. All right, and here we have some examples. So the plate farthest to the left has been streaked with Staphylococcus aureus. It's gram positive. So the whole surface of the plate is covered with Staphylococcus aureus. The middle is E. coli, which is gram negative. And on the right, you have Pseudomonas aeruginosa, which is another gram negative bacterium. Onto each of these plates that are covered with bacterial growth have been placed discs. So they're paper discs that have been soaked in different chemicals. So you see chlorine at 12 o'clock on each of the plates. And you'll notice around chlorine, there's a clear area. And that's called the zone of inhibition. And basically, that clear zone is where there's media, but no bacteria are living there. So chlorine is a pretty good biocide for these bacteria. If you look at phenol at 3 o'clock, we see zones of inhibition for Staph aureus and E. coli, but around Pseudomonas, there's hardly any zone inhibition at all. At 6 o'clock, there's a quat microbicide, and we see a large zone of inhibition for the gram-positive bacteria, a small zone of inhibition for the E. coli, and again, I don't think there's anything around the other gram-negative. And if you look at 9 o'clock, hexachlorophene has a large zone of inhibition for Staphylococcus aureus, but for E. coli and Pseudomonas aeruginosa, the gram-negatives, it seems to have no effect on their growth. So using these treated discs is a way that we can test for resistance to these different microbial controls. All right, let's talk a little more about prions. These infectious proteins are known to be heat and protease resistant. Protease is a chemical that breaks down proteins. Autoclave, if we use an autoclave and sodium hydroxide, so we combine that high temperature and pressure along with a strong base, that is effective at destroying prions, or incineration. You can burn them. All right, some important things to keep in mind, though, about chemical disinfection is there's no single disinfectant that's appropriate for all circumstances. They affect microbe cell walls, cytoplasmic membranes, proteins, or DNA, and the chemical compounds are not to be used to treat disease. These are only for controlling growth and preventing disease. So how do we clean things to help keep people healthy? All right, so some other methods, chemical methods for microbial control are the use of antimicrobial soap. Is this too much of a good thing? And how it's led to the development of resistant microbes. So there's little evidence that products containing antiseptic or disinfecting chemicals add to any human or animal health benefits. Use of antimicrobial soap should be limited to food handlers, people caring for newborns, or people caring for high-risk patients like immunocompromised people. The CDC recommends using regular soap and washing with warm water for at least 10 to 15 seconds. There's little evidence that products containing antiseptic or disinfecting chemicals add to human or animal health. And I'm talking about things like hand lotions and hand soaps and places where they're not really needed. The use of such products promotes the development of resistant microbes, and a good example of this is triclosan. Triclosan has the ability to disrupt plasma membranes. It's an active ingredient in antimicrobial soaps, toothpaste, deodorant, and it's incorporated even into cutting boards and knife handles. Widespread use of triclosan has resulted in reports of resistance. So I know Colgate used to have triclosan in it, and they changed their formulation in the last few years so it no longer contains triclosan. But it would be interesting to look through your own bathroom cupboards and cabinets, look and see if you have any products that contain triclosan, and think about whether or not that triclosan adds any value in improving your health. All right, and that's it for Chapter 7.